So just before I get started here, I just mentioned to a couple of you, I changed this whole thing about 2 days ago. I mean, I started working on this stuff in August. And I didn't, you know, I called one of the prizes, but I was pretty close on the other two. But it's what happens when you get into stuff and you're usually close to the end and you realize that it's really not what you want to say, or more commonly, I've found that when I've been writing papers or writing books, if there's a writer's block, it usually means I don't understand the stuff. And stop messing around with the writing and get back to the stuff and figure out what it is that I don't understand to be able to write it. And that certainly comes up with those little lake report things, because you're forced to summarize something in kind of less than 1,000 words. And it's hard sometimes to get stuff right. And I'm actually going to use one of those articles in a few minutes, because you fiddle around with the stuff until it actually, you do understand it enough to be able to write it. Doesn't mean that somebody else is going to be able to understand it from what you write, but at least I'm ahead. But it fits with, remember the business on the chemistry prize last week, and AlphaFold, and they had done Alpha 1, and Alpha 1 really worked really well. It was better than anyone else's program, but not good enough for the team, and they trashed the whole thing and started over again. And as I mentioned last week, it takes a lot of courage to do something like that, particularly when you've invested so much of yourself and the team, and in some cases money, to do that, but they did it. And then they really crashed it. They really did a great job. Oh, good.
Sir,
Yeah, come on in.
nice to see you.
Yeah, good to
No words over 6 letters, okay?
Yeah, okay, Ross, all right. Yeah, okay. That's 6 letters is the limit?
That's my max.
Oh, I see, okay. Well, let's get started. So, hi, Jane. Good afternoon and welcome to this, the last one. So this is the Medicine Prize, which is sometimes for me the most challenging, has the broadest range. But there is some perspective in this. I did give a series some years ago on the Camelot period in physics in 1900 to 1930. You could stretch that to 1940, or at least the beginning of World War II, when budgets were small or non-existent and people were really brilliant, and there weren't many of them. And the quality was superb. Well, biology, when I look back now, and I didn't hear about this in high school or in university or even in my postgraduate training for probably 20 or 30 years. But with 1953, with Watson and Crick, that profoundly changed the roadmap. Because up to that time, biochemistry was biochemistry, and biochemistry was all about the architecture of molecules. But how it changed to now coding. Coding was the key, information and coding. That's what's in DNA, that's what's in RNA, that's the final product in proteins. And that's where the whole emphasis changed to. And there was another magical period of about 30 years, 40 years, and the equivalent to that Camelot period, but happened in molecular biology. And it continues to this day, and the laureates we're hearing about today, and their fathers, the ones who mentored them, and the preceding ones before that. I mean, there are probably only 5 generations, 6 generations that we're talking about of laying on of hands here. And people can trace their background. They actually know somebody who knew Crick or Bringer, and may have met him. And so that was very much in play. And it's also a period when physics, which always was out kind of out there, the strange world of physics that we started the 1900s with. And it all seemed fairly irrelevant. It's anything but irrelevant now. It's very relevant to artificial intelligence. It is certainly very relevant to DNA. We heard that last week with the structure of proteins. That's what the enhancement was to the Alpha-2 program, to acknowledge that physical principles at the atomic level made a huge difference. So things, perspectives kind of come together here. Now budgets are larger now, groups are larger, they kind of multiply, but in the world of ideas and fresh ones, it's still alive, very, very much in biology. So I wanted to bring that to your attention. The other thing that struck me was that, I do write these articles for the Advance, and I don't know whether this one's come out yet or not, but I wrote one on From Worm to 8 Nobel Laureates. Well, of course, that's C. elegans, the worm that we're talking about today. And it's just been such a fruitful period in biology. But I think I phrased it well when I wrote the article, so I'm just going to read a little bit. I don't like reading stuff up here, but I'm going to do it now. And this year's Nobel Prize in Medicine celebrates another big step toward understanding a mystery in biology. How do cells carrying the full suite of genetic instructions manage to differentiate, branch into many hundreds, if not thousands, of different appearing and functioning cells, but they have the same genome? How does that work? Yet that is precisely what young cells do, beginning with the fertilized egg, part of which develops into the placenta, and the other part which develops into the body, step by step, developing into cells that take on different jobs and appearances, but with the same genome. The answer is that, well, the same genomes are capable of directing cells from different developmental paths to, say, certain cells in the cerebellum in the brain. They must also be capable of turning off, silencing, whole suites of genes that are related to many other cells in the genome's repertoire. So you're increasingly turning on more function-specific genes, but you're turning off a whole lot of other genes. That's what's going on. And you're also retaining the genes that are in common to all cells. So you're not dumping everything overboard. You're only talking about those genes that relate to specific functions of a certain cell. And then I went on to say, and it's very relevant to today, that may have been the working hypothesis, but how do scientists prove it? The answer is often to turn to simple biological principles. And this isn't often stated, but this is true. Banking on the now well-established proposition that the kingdom of all life, while varying wildly in how its members look and behave, goes about its business by sharing many proven genetic solutions to life's diverse challenges across the broad spectrum of evolution's life forms. That is a key statement to make. That is absolutely key. And so that's the assumption that scientists use when they choose a simple animal like the nematode we're going to use today, or they used. But it happens to be true. And it takes you back in evolutionary history, as this stuff that you're going to hear about today takes it back 600 billion years, more, or maybe more than 1 billion years, to before plants and animals diverged. It's hard to think of a time before plants and animals, but there was a time. And it was well over 1 billion years ago. So that's what this is about. So With that perspective in mind, we will go ahead. Well, the usual pictures of the Pelosi, there they are, Ambrose, Victor Ambrose, and Gary Ruvkin. Interestingly enough, they're within a year apart at their birth. They're good friends, have been good friends for many years. They worked in labs, one above the other. They had the same supervisor, Horowitz, a Nobel Prize winner with Brenner. So you can see the laying on of hands taking place here. And Victor Ambrose is sometimes committees, promotion committees at universities, particularly Harvard, don't get it right. This pair had just solved the reason why they got this Nobel Prize, and they turned him down for tenure. True enough, the implications of the importance of what they had done wasn't widely appreciated. True. But looking back, committees make mistakes. So move along. So, sorry, we don't want to go back to the beginning. Well, here's creating Nobel Prizes. Pay attention, read it. Here, if you have a lot of money and a bright idea for a new prize, perhaps in mathematics, AI, psychology, paper dresses, or writing habit, make your proposal to the Norwegian Academy of Sciences for a new prize to be administered by the NAS. Once your many millions is deposited in a Norwegian bank and you promise never to interfere, we will create a bust of you whose size will reflect your contribution and our gratitude for funding our retirement. So there we are. That's how the economics prize, I mean, this is a joke, but it's how the economics prize is created. And I think they're looking at expanding the prizes. Sorry, why am I way backwards? Because I'm pressing the wrong button. Well, here we are, the scientific background for the discovery of microRNA. I mean, so that's the prize, Victor and Victor. Interesting first names, right? Yeah. Oh, one of them's wrong, isn't it? So, oh, okay, back there. Well, here we are. I take issue with the artist. This is always the same artist for as long as I've been doing this, doing the portraits. The hair always looks the same. And I mean, he or she certainly captures the image, but there they are within a year of one another. And well, here's the thing. Here's a DNA, the genome here, and part of that gene transcribes. Well, all of DNA transcribes to messenger RNA to protein. That is the central core of modern day molecular biology. And part of that genome might end up as part of the brain, and another part of muscle, or lining cells in the gut, or bone, or whatever it is. But it's all about coding. And so, I've already mentioned this, but here are some examples where simple systems really work. Andrew Huxley and Alan Hodgkin were a pair from at Cambridge University, and in the late 1930s were investigating or very interested in, how cells transmit electrical signals from one cell to another or along a single nerve fiber. And the equipment of those days, I mean, they had to build all their own stuff, even in the post-war period. They had no budget. There were no grants. No one had any money. So they built all their own oscilloscopes, their stimulators, you name it. And they had to fix them too when they didn't work. And two of them were brilliant, and they got back to it in 1945, 46. And they are amongst the most meticulous studies I know of. They are beautiful to look at. But they used a giant squid axon, so large that they could actually get their electrode inside the axon. And so it was an easy model to do. And all of the findings have turned out to be true and applicable to us from a squid. So that again reflects this this commonness in nature. You solve a solution. If it works, nature retains it. And then Eric Kandel, a fascinating guy, just got out before the Nazis took over Austria, and I think he lost several family members. Became a psychiatrist at Harvard, and then saw his true calling as a physiologist. And he did a study of memory and conditioning, and got a Nobel Prize for it. And very clever guy in the arts too. Has written a lot about the relationship between neuroscience and art, and it's worth reading because he writes well. Well, we end up with C. elegans. I like the last name. It is really elegant. To study programmed cell death and the genetics of organ development. They got a Nobel Prize here, but I'll point out that I'm going to say something about Sidney Brenner later. Robert Horvitz was the mentor of the two laureates we're celebrating today. And John Salzman, when you see their pictures, they are nice guys, classy guys, all three of them. So if you could say that these two are related to Robert, and in a way, they are. Sidney Brenner introduced this nematode, this C. elegans. I mean, there are lots of different types of nematodes, but he chose this particular one. And what are they looking for? An organism with enough complexity, but not too much, that it gets in the way. That's easy to work with. Well, why is C. elegans so great? It's small, it doesn't live long. It does have some organ systems, but it certainly doesn't have a complete set. And you can see through it, and you can see and count every single cell in it. And you know what the connections are. There are precisely 302 nerve cells, precisely, every time. So it's an ideal organism to work on. It's great. So they got a Nobel Prize, the three of them, back in 2002. Well, there it is. There is the elegans, and so here's what's it got? What does a minimalist organism have? Well, it needs nutrition, so it needs a gut. So we have a pharynx, an intestine, something to convert whatever it eats into energy. And something to make babies. It needs gonads. Most of these, 99% of C. elegans go both ways. They create sperm and ova, and fertilize themselves. But otherwise, basically, really very, very simple. But the beauty is, it's all in the open. You can see it and count it. And again, simple enough to see. If the animal's too simple, then if there are mutations in genes, how would you ever know it? Because you have to see it expressed in something lost or changed. And yet despite it, this is staggering to me, it has as many genes as we do. Humans have somewhere between 20 and 25,000 protein encoding genes. Apparently, that little thing that's a millimeter long has over 20,000 protein encoding genes, and 35% of those genes, a third of them, are homologous to equivalent genes in us. So there's a nematode in us. No. Okay, and not much of a nervous system. Well, what can you do with 302 nerve cells? Not a lot. What's missing? No heart, no circulatory system, no immune system, no eyes, but interestingly enough, the photoreceptor proteins are 10 to 100 times more sensitive than ours. So in the muck, whatever it's in, it's good at picking up any form of light and better than us. Well, here's what all of that, well, here we are. So the 8 laureates, I've mentioned some of the stuff. Oh, you know, there are in-groups, and one of the most in-groups in biology for the last several decades has been the C. elegans group. I mean, they get together every 2 years. It's as much a social as a scientific meeting. It's a small club and that gets together. It's growing, obviously, with the generations, and they have these international worm meetings. They have their own journal, and so it's I mean, that creates the bonding in science, doesn't it? When people have connections with one another, particularly when the groups are relatively small, they form close relationships, and they share with one another. That's not true of large science groups, or many of them. I love this picture. First of all, it's black and white from the Nobel thing, so it's that old. So this will go back to 2002, the year they won the Nobel Prize. From what I've read about the 3 of them, they are all a class act. This is the one that kind of looked after or supervised our 2 laureates that we're talking about today. Horvitz at Harvard. Sidney Brenner, I'm going to say more about later, not so much John, but not because he's not interesting. This fellow has a very interesting life story. I'm going to go back to this programmed cell death. I don't know how much you know about programmed cell death, but I mentioned this in another series, but in development, the brain creates many more nerve cells than it's ever going to retain. And there's a certain competition that takes place. It creates maybe 2 to 3 times the number that it's going to retain. And through a competitive winnowing out, meaning whoever makes the strongest connections lives, and the others disappear without leaving a trace. So that's programmed cell death. So from the moment almost that we're born, we are already, death is, if you like, death of cells is beginning. We think of death as the day we have our heart attack, but it isn't. It's a lifetime process. We're losing billions of red blood cells and immune cells and all that, skin cells and that kind of thing. So it's a long, long process to which the final act, the very brief part, is only the final act of a long program. Anyway, so programmed cell death and it's things like it go back a long time. Well, this is what I mentioned kind of earlier. I mean, I mentioned this dilemma in the article in the paper whenever it gets in, and but this dilemma of, gee, you have a full set of genes. How can you differentiate into different cells? Well, we talked a little bit about this, and the key is this right here, that as cells differentiate and branch and become more and more what they're finally going to become, it chooses more and more very specific and relevant genes, and it turns off all the others that it doesn't need. Okay, that's what I was saying earlier. And how does that take place? Well, we thought we had the answer to that maybe 15 years ago, because the genome has I won't call them police, but there are proteins that can turn on and off genes, and some RNAs that can turn off genes directly. What they discovered, and why they got the prize, is because there's an entirely different system that silences the impact of genes, not by silencing the gene, but by silencing the messenger RNA. Remember the basic process in biology? DNA, messenger RNA, protein. That's the central dogma of all of biology. Okay, so you can either silence the gene directly, which we knew about before these people did their work, and we knew a lot about it. But what they showed is that there's another end run around directly silencing the gene. Why not silence the messenger RNA? One step further along in the process. You follow what I'm saying? Okay, and if you do that, the protein isn't translated. There's no protein. I got it almost given the talk before we're a quarter of the way through it. So, so we'll just move along. Here. Okay, so, well, what about, this is a key thing. You've got to find some genes, and you need to clone the genes. You have to identify them. And in the 1970s, Brenner's lab uncovered one mutant gene variant, which they called LIN4. And it's striking. One gene, in its mutant form, these worms displayed a striking phenotype. Many cell types and structural traits were entirely gone, and eggs accumulated because the vulva failed to develop, so they couldn't get rid of the eggs. So you had these big nematodes stuffed with eggs with nowhere to go. Boy, that's a lot of impact for one gene, which tells you that this is a very special gene. This is a kind of the emperor gene. It has a broader impact. Okay, and then they later found that so that first one was called LIN4, and then they found another one, LIN14. So where are we here? So, and and they do look a lot different. Here's the normal-looking nematode. Here's the one jam-packed with ova, with nowhere to go because there's no cloaca, there's no hole to get out. And so that's the impact of one mutant gene on this nematode. And here's the impact of another gene, mutant gene, 14. It's all kind of shriveled up, and it doesn't have any ova at all. And it's really missing. I mean, there aren't many systems here, and half of them aren't there. So it's a huge impact for the nematode. So that gives them a very good target, doesn't it? It gives them two genes that they know a lot about, the impact of those genes, that they can study. So you don't have to investigate 20,000 genes. You can investigate two that you characterize very well. Well, this is the key slide. This is the bingo one. Okay, so just follow me through this here. In the lab, remember Horvitz is the supervisor, Ambrose and Ruvkin each have their own labs, but it's under Horvitz, the one who worked with Brenner. Began their long pursuit. What's long? Years. Several years. Took a lot of work doing this. And in trying to clone, it's much easier to do this kind of thing these days because the technology is a lot better. But then it was really difficult. But Ambrose, these two men got along quite well. They talked together at the end of the day, they exchanged their news, families, that kind of stuff, but they also talked about their work. They actually cloned the same genes independently, which is a good thing to do. And so here we are, a very specific evening, the evening of June the 11th. This is where the Nobel Prize was born. On 1992, Ambrose and Röntgen exchanged. What they did is they had sequenced the genome for these two genes and the RNA for the Lin14, and also for the micro. Oh, I didn't mention this. The Lin4 gene had one peculiarity which they'd never noticed before, and that was it didn't code for a protein. It coded for a small snippet of RNA. And for a long time they thought, oh, well, you know, RNA is a very fragile kind of thing, breaks up, fragments easily, it could all be artifact. But after a lot of laborious work, it was obvious it made a really small little RNA molecule, and it was the same one, and it held up. So that's the information that they had. But then they had sequenced the genome, or the sequence of bases, right? And then they compared notes, and wow, there's the eureka little light coming on, because two separate genes, and they found that the Lin4, its little small RNA piece, the sequence of its bases was very similar to the sequence of bases in the RNA created by Lin14. So in fact, this is post-nuclear interference, if you like, or a connection between the two. So it meant that the Lin14 was regulated, the effect of the Lin14 gene was regulated at the post-transcriptional level, beyond the nucleus. And its impact was on this, on the messenger RNA of Lin14. That's the key, because now they knew that the whole new world. They had found another way of modifying, or in this case, silencing this particular gene, but not through acting on it directly, but by acting on it on its messenger RNA. It's a kind of a bypass. Do you follow what I'm saying? Because it's really important, because that's the core. That's why they got the prize. Well, they were very excited, but no one else was. They published the papers, no one responded to anything, and 7 years went by. And the reason for the silence was, people looked at this and said, oh, you know, that's just C. elegans, you know, it's heaven knows, it's, we believe what the guys did. Technically, they did a good job. The stuff is as they say it is, but it doesn't apply to any other animal. Until they found another. Well, here we're just in diagrammatic form, but here we are. Here's the second. They found another microRNA, which they called Let7. But the big thing about Let7 versus the Lin4 gene is that Lin4 gene was only in common among nematodes, different nematodes. But here's Let7. We have Let7. Other mammals have. And it's widely spread throughout the animal kingdom. So it's been conserved and spread and probably had a very early start. Anything that had that early a start and is so widely conserved is obviously important, or it would not have been conserved. So that really opened people's eyes, and they said, oh my God. And then the gold rush was on for looking for these microRNAs, because here was another way of editing the genome, which no one had guessed at absolutely before. And it was highly conserved. If it was highly conserved, nature liked it. It worked. Otherwise, it would have been disposed of. So wrapping up, the collective evidence became compelling. A vast class of regulatory microRNAs existed across a wide range of animals. Counting all species tested so far, they found 49,000 plus of these things. Now here's the thing that boggles my mind. MicroDNAs may have emerged multiple times during evolution, including the early lineage of multicellular animals. Well, multicellular animals didn't really evolve until maybe 500 million years ago. Life has been around for over 3.7 billion years, but it's only in the last 500 million that it became complex, meaning multicellular. Okay, and if you go back further, the hints that said hinted at earlier, if in fact this type of DNA influence antedated the split of plants from animals, wow, that takes it as far back as it goes, almost after the appearance of complex single cells. So it has a long heritage and has been held onto. So that's why they got the prize. I mean, they got the prize for two things: discovering this post kind of nuclear gene influence, and showing that there could be there was another process, and probably a very important one. But it has such a deep history. So if it has a very deep history, well, it evolved very early in the game. So and this, the Nobel Committee, they come up with these kind of their artist drawings, but this is 500 million years ago. But plants, you could move this back another 500 million years, go back to 1 billion years ago. It's ordered, so a long time. So it's interesting. They had no idea of this when they did this. They were just curious about these two mutant genes and what about their business of being mutant, and what the mechanisms were of silencing these genes, and literally stumbled on a mechanism to explain it, and with huge implications, completely unanticipated. No one saw this one coming. And it's really stunning. So this is, sometimes I like the committee to speak for themselves. Thanks to the seminal discoveries of them, and they have a certain arcane way of talking about this kind of thing, but they do, this is the thing. This unexpected layer of post-transcriptional gene regulation has critical importance throughout animal development and in animal cell types, and is essential for complex multicellular life. That's it. Nicely captured by them. And well, I've already said something about these two gentlemen. What impressed me in learning about them is that they were so collegial, they worked in labs that are close by. You know, at places like Harvard, I'm not singling out Harvard in this regard, but they're highly competitive. People don't share stuff. It's a high stakes game getting grants. And so the tendency is not to share stuff if there's any chance of it risking your career. But these two were quite different. And I think they were like that anyway, but their mentors were that way. And that's why I think this club, this Worm Club, gets along so well and has such a long heritage. They're actually closely knit with one another, so that's a good thing to know. Lessons learned, well, lots of lessons to be learned. A lot of these apply to almost anything we do. Success, well, success depends on chance. After all, a lot of people don't have a chance. If you're living in 3/4 of the world, you don't have a chance to do any of this kind of stuff. You never will have. And schools have to be good, you have to have the opportunity, training, discipline, all of those things are important. You also have to know when you hit it big. And they did. That date that I mentioned, that evening, they knew that they had hit the jackpot. They had done something that no one else had done. And a sense of humor about it, and that seems to run through the group. Sidney Brenner, a little bit about him. He was born in South Africa, but the parents were from Eastern Europe. The father came over first, then the mother. He was a cobbler. I think she cleaned houses. The kid was precocious, got into medical school at 15. He didn't mess around, but you know, the graduate opportunities in South Africa were not great in that time. And the family certainly didn't have money or influence, but he got his DPhil at Oxon at 24, and he and Crick hit it off. I'm not surprised. I mean, both men quite outgoing, both very highly intelligent, both loved discussion, usually over wine or beer, and pubs, and that kind of thing. And I like his sense of humor here. Title is Scientific Talk. This is at the Nobel thing. Nature's Gift to Science. Well, it's his tribute to C. Elegans, which I thought is kind of a great thing. He's known for his wit, or was, he died a few years ago, for his wit and insight, again, like Crick. This is you wouldn't know this, but this journal is a high-ranking journal. This is like the Lake Report, boosted up, and this guy gets to write his own little thing with every issue. And it was wonderful stuff. It's absolutely wonderful stuff. If you read the book, The Eighth Day of Creation, it's a bit of hard slogging, but if you do, that kind of fine-grained detail comes out about some of these people. I could imagine he would be very, very good company anywhere, in almost anything. 2 species are named after him, other than worms. And so an interesting guy. He, well, I like this one. I like this one because this is his speech at the, they've had a dinner, the celebration, the things have all been awarded, and there's been a celebration. So just bear with me. And so, Your Majesty's Royal Highnesses, ladies and gentlemen, after that speech, first of all, look how short it is. I'm afraid I come as an anticlimax. I was asked to prepare my speech because I understand it will be captioned for the Swedish public. And so it will be difficult for me to change what I have to say because That will be very confusing. However, I will add something which arises from what we have seen in the brilliant show, which you can't see, that was presented to us, because it gave me many ideas for a uniform. This is very plain. A uniform for Nobel laureates. In particular, I like the person who had such a swollen head that it could be easily converted into the bubble economy of the time. And for my own choice, if I had any hair at all, which you didn't, I would dye it orange and put on a gold lame dress and wear a top hat. Okay, that's his little thing. But this is great advice for any graduate student. And now I come to what I want to say, and the very best way I can say it is to tell you about a letter I have received. A Nobel Prize winner gets many letters, yes they do. This was from a student in China. His email said, Dr. Sidney Brenner, I wish also to win a Nobel Prize. Please tell me how to do it. So we now, in the next 8 lines, have how to do it. All right, this is your kit. I had been considering the reply, which will say something like this. First, you must choose the right place for your work with generous sponsors to support you. Cambridge and the Medical Research Council will do. Perfect. That's where he was for a long time. That's where Crick was. Absolutely. Then you will need to discover the right animal to work on. A worm such as C. elegans will do, for example. Next, choose excellent colleagues who are willing to join you in the hard work you will need to do. How about John Solston and Robert Horwitz? Well, those were his 2 co-laureates. For a starter, you must also make sure that they can find other colleagues and students. This is very important. I had a friend of mine, and he's retired at McMaster, but he knew one laureate, and he was in his lab for several months and was becoming a little disgruntled because he didn't see much of him. And then they caught each other on the tube one evening, and he said, well, can I talk to you? Other than tea in the morning and afternoon? Oh yeah, come along. So he said he was frustrated, but then Huxley said to him, well, you know, we need to know whether you can sink or swim. If you can't make your way, if you can't find colleagues to work with, if you can't come up with ideas yourself, it's probably not worth your while being here, and it's not worth our while keeping you here. That was just a very honest appraisal of a high-class place and a very productive one. As it turned out, the fellow I'm referring to was productive. He was actually doing those things, but it was nice to hear it said. And not everyone would take that the way I do, or maybe, so I think he was handled properly in the lab. Sounds harsh, but I think it's right. And then I love this last line. This is about the 3rd time he talks about working hard. Anyone that ever had anything to do with Brenner or Crick worked hard. And then all of this is necessary. Oh, here we are. Everyone will have to work hard. Finally, and most important of all, you must select a Nobel committee which is enlightened and appreciative and has an excellent chairman with unquestioned discernment. All this is necessary to bring you to this point where, on behalf of your colleagues, you can thank everyone for the opportunity to be present here and to make this speech. Isn't that a lovely way of doing it? Yeah. It's very classy. And so how are we on time? So just a little bit. I want you to pay attention to this too, because Crick was a very interesting guy. I mean, you can read it yourself, but I think this is a very good, this is from the book, The Eighth Day of Creation, by somebody who knew him well, but others would share this. The one angle of view that commanded a consistent, clear perspective of the next complexities of biology was Crick's. He thought up many of the developments or stimulated them. Most of what he did not originate, he soon became aware of. Besides the talent for theory for which he was known, he had a talent for friendship in science that quickly grew into a leadership role. By brain, wit, vigor of personality, strength of voice, intellectual charm, and scorn, a lot of travel and ceaseless letter writing, Crick coordinated the research of many other biologists. And this is a really important one. Disciplined their thinking, arbitrated their conflicts, communicated and explained their results. That's saying something. And as he went, he sorted the important from the less important with a brisk efficiency that now serves well to distinguish the main line of molecular biology from all else that was happening in biochemistry. He had an outsized influence, more than Brenner. It's that he's really acting as a node, isn't he, of cajoling people, encouraging people, getting in to know himself, helping people understand actually what they're doing and how maybe to do it better, and in some cases actually recognize what they've done or could do, and bringing out the best in people. I think that's what more can you ask for? I'm not going to get into this stuff at all, because that's a whole other, there's a whole other parallel in this period. I mean, this may have been a long Camelot period in molecular biology, say from the mid-1900s through to the beginning of this century. But there was another parallel track to what we've talked about here. And the parallel track was the studying stuff of these two people who I'll go back here, of kind of reverse engineering cells, taking adult cells, implanting them in an embryo, and creating an animal. You think about it, no one would have thought that in the 1700s, 1800s. I mean, that's a stunning thing to do, and get away with it, and show that it's possible. And then take another step, and take those cells that had become highly specialized. I mean, the first 9/10 of this talk was about specializing cells, the mechanisms, kind of silencing genes. Here, they're taking all those little tags off and restoring the potential of all of those genes so that they can actually create a whole organism or a whole organ. That's remarkable. That's current biology in the medical sphere now too, right? Restoring people's bone marrow, early efforts at growing kidneys and organs like that. So biology has really moved along very, very quickly, and these two guys were leaders in that. So I'm not getting into that. So I'm going to cut it short now, because we've talked a lot about it. These three talks actually fit together well, don't they? Because the first two were really all about AI. The first one chiefly about, well, what is AI and how does it work? And the second one was applied AI to proteins. And then, as I mentioned at the beginning here, this talk is really about information, biological information, and coding, very similar to what's happening on the electronic computer side. I mean, there are parallels between these, so they're actually related. And I think so. So the three prizes dovetail. I think the sciences, as this is now the 8th year, are coming closer and closer. What were separate islands of quantum mechanics and astrophysics and biology, and they're very separate. And we know that every university has different buildings for them, and a whole bunch of teachers teaching that particular subject. But who the hell actually pulls it together? But I think the Nobel series this year and the last couple of years is beginning to try to pull that stuff together. And the science warrants it. I mean, I don't think you could do protein studies today without understanding stuff at the atomic level. That was the lesson of John Jumper and the AlphaFold. You can't do it. When they introduced that, the stuff really took off. Before they introduced it, they were very good, but they weren't excellent. And he had the guts to say, no, wipe it clean, start all over again, but this time we're going to pay attention to the physicists. So I think this year has been very interesting, but it does fit into a pattern for if you think back, at least to the science ones, last year we talked, two of them were about electrons. And so that's deep physics, or we call it physics. It's just another dimension at another level. And that's a good way to think about it. So any thoughts or questions? I'm not going to get to prions. No? Okay. So, yes?
When the first person was denied tenure at Harvard, did he ever go back to university teaching?
Sorry, who did?
The first person you mentioned was denied tenure at Harvard after he made his discovery, which was highlighted?
Oh, yeah, no, he went back to teaching, and he ended up in University of Massachusetts, Western Mass. But he didn't get tenure at Harvard. Harvard is notoriously difficult to get tenure at. And you know, I don't know if you've ever sat on committees that do that. It can be very picky, and particularly when personalities get involved, and people are very competitive.
Was that a hospital, was that Western Mass, or? Is that a hospital?
It's a university.
Western Mass?
Yeah. They have a university attached. Yeah. Okay. I think he would have liked to have stayed at Harvard. I don't think he has any grudges. There's nothing he's ever said that I know about. And the two men, remember, they continue to get along, and the families know one another, and they do stuff together. So that tells you something. So, all right. So, economics prize next week, a tough one, David. And I think it's very tough because this stuff is relatively easy, but there are so many factors involved in economics that how the hell do you ever get your arms around it all? But David does that successfully every year, so next time.
Yeah, just a little bit about that one, the next one. This, the economics president was interesting this time because the first economist was Adam Smith, and he wrote something called The Wealth of Nations, which we're all familiar with. It's about wealth. And I don't know if this says anything about where we're going with economics, but there are three authors. All of them are from MIT. They're economists, and their book is called Why Nations Fail. So we've gone in 200 years from the wealth of nations to why nations fail. But it's not as tragic as it sounds. It has a lot to do, as you can imagine, with politics and the people who are running the various countries. Certain countries succeed fabulously, and not for physical reasons, not because they have better geography or better minds or whatever, but for often, at least these authors contend, for the political situation that they are working in.
We're just trying to get free time. Okay, so next week economics, and then Peace Prize, and then Literature Prize with Valmai. So that's it. So far, I think it's been a good year, and maybe it gets better, right?
Thank you,
Okay.
Bill.